So our next speaker is Kristen Nelson. Kristen earned a BS in Environmental Management and MS in Biology from Cal Poly San Luis Obispo. Kristen is the CNPS Rare Plant Program Manager, where she oversees and coordinates field-based projects, including various community science and volunteer-driven projects. She's currently supervising a master's project modeling the habitat niche of San Luis Obispo fountain thistle, as well as a project, the projected range under various climate change scenarios for that species. She's also a co-author on the soon-to-be-published paper on the distribution and quality of western monarch butterfly habitat across California. So without further ado, Kristen.
All right, good afternoon. We're almost here at the end. So I'm going to be talking to you today about a project that was completed in 2024. This work was focused on both seed and data collection for 3 rare species that were impacted by the 2021 Caldor fire in Eldorado County. This work was led by CNPS staff in coordination and funded by the Forest Service and made possible by a pretty large cohort of some really excellent volunteers that helped us complete this work. Maybe a little delay. I might have a slide jump here. There's a lot about this project that I'm not gonna have time to talk about today, but I am gonna just provide a little bit of context. And because we're botanists, I am gonna at least touch on and introduce all 3 taxa that we targeted as part of this work, but I am gonna have a little bit of a focus on just one of them, which I have bolded there, the Sierra Valley or Hutchison's Lewisia. So, starting with a little bit of context. I don't know if the battery's dying or if there's a magic trick here, but do I point it at the computer?
Hey, is it this one?
Okay, thank you. So I do want to start with a little bit of context, and very briefly before I dive right into discussing the interesting and frankly very fun work that we got to do in this post-fire landscape, I wanted to just briefly take a moment to acknowledge that this was a pretty high-intensity fire that was pretty devastating and traumatic for the local community. Throughout its footprint, a lot of the area, as I mentioned, was pretty high intensity and severe fire burn intensity, and in the end, more than 1,000 structures were damaged or completely destroyed. And especially this week, as the devastating events are unfolding in Southern California, I just didn't want to lose sight of that reality of fire, and there's a lot of cool work to be done, but it also has very real impacts in these communities. So just to orient us very briefly, this fire burned for a little bit more than 2 months between August and October in 2021. Its final footprint was just shy of 222,000 acres. It was almost entirely contained within Eldorado County, but it did creep a little bit into both Alpine and Amador counties. In this map here, I'm just showing an aerial image of the area where the fire occurred. For context, up there on the right side of the image, you can see the southern part of Lake Tahoe, and in the far lower left of my image, that is Folsom Lake, so just for context and orientation. I have 2 fire scar boundaries here, and they might look familiar if you remember from Ed's talk yesterday with Comstock seeds. The fire boundary outlined in heavy red is the Caldor fire, and up toward the top of my map there is the mosquito fire footprint. And then the bright green overlay is just all of the Forest Service managed lands in the Eldorado National Forest. So we had a few project objectives. First and foremost was to collect seed for our 3 target rare taxa. These species were actually selected by the Forest Service as priorities for seed collection with this fire recovery money. And one thing that we wanted to accomplish was 1 seed collection for each of the taxa that would go into long-term seed banks, so conservation seed banking, and then an additional 3 collections that were returned to the Forest Service, additional 3 collections for each taxon that were returned to the Forest Service for more short-term objectives like enhancing and restoring impacted populations. Second objective was also, of course, we're out there, we have boots on the ground, we're visiting these populations, we want to collect good population data, as well as herbarium vouchers. So we did that while we were out there as well. And then our last objective, and this was a fun part of the project, was that we actually had a stated project objective to do a pretty significant volunteer engagement effort with a stated goal of 425 volunteer hours. So here's the fun part. I'm going to go ahead and introduce all three of our target taxa fairly briefly. The first one here is Navarretia prolifera subspecies lutea, the yellow burr Navarretia. This taxon has a CRPR4 rank of 4.3, indicating that it has a limited range, but may be common where it does occur. And in fact, it is endemic to Eldorado County, so definitely a very narrow range. Our second target taxa was Calochortus clavatus variety avius, the Pleasant Valley Mariposa lily, a real stunner if you have not seen it in person. This species has a CRPR ranking of 1B.2, so indicating a fairly rare and endangered species. And it spans a relatively narrow elevation band here across these three foothill counties, Eldorado, Amador, and Calaveras. And then last, but absolutely not least of all, our last target was Loesia kellogii subspecies hutchinsonii. Don't add an N to that. It's not Hutchinsonii. This is the Sierra Valley or Hutchison's Loesia. And as currently circumscribed, this species is recognized as having a range that comes all the way up here to the northern part of the state with some disjunct populations down here through Shasta County and all the way through these foothill counties as far south as Tuolumne County. But it's worth noting that this one has a CRPR rank of 3.2, indicating that we recognize some taxonomic issues and ambiguity within whatever is currently circumscribed as and recognized as Loesia kellogii subspecies hutchinsonii. And on that note, I just wanted to say that there is a forthcoming new species that has been confirmed, and I believe it was talked about at last year's NCB conference, and that Loesia is a segregate of Loesia kellogii. So certainly some work to be done here for anybody that wants to pick this up. Okay, and this part of my talk, I'm going to have to zoom by. I wish I had more time to talk about the volunteer engagement. There was quite a bit of logistics and work that went into this. We ended up with almost 60 volunteers, which was frankly way more than I could have hoped for or dreamed of. And they were such a wonderful group to work with. But organizing 60 volunteers to do this kind of work does take some structure, some logistics, some planning for coordination and how they're going to collect their data and how we're going to QC the data, et cetera. So I just wanted to mention it because if anybody has interest in hearing a little bit more about those logistics, feel free to reach out to me via email later. And just very briefly, I'm going to go ahead and note I think it was a wildly successful effort. So here in the good news, in 7 short months, our volunteers logged 126 workdays, just shy of 500 hours, and more than 3,800 driving miles in support of this project. And in combination with our staff efforts, we submitted more than 150 survey forms, so that's just data collection forms, collecting data on all these populations, and visited 58 distinct population groupings across all 3 taxa. So again, follow up with me if you want to hear more about the volunteer coordination effort. I'm briefly going to share some of the results from the Navarretia and the Calochortus, and then talk a little bit more in depth about the Loesia. So starting with the Navarretia, the seed collection process and method for this taxon was pretty straightforward and fairly typical of small annual species. We essentially just collected entire plants. Once they had senesced and dried out pretty thoroughly for the season and the seed was well developed and mature, we collected whole plants and put them into envelopes. We ended up with a pretty high number of vouchers for this one, so 12 vouchers from 12 unique populations and almost 43,000 seeds from 279 maternal lines. And that's inclusive of seeds that went to the seed bank and ones that went to Forest Service, so that's just all lumped together. One of our primary challenges with the Navarretia was relatively straightforward, but in portions of its range in the Eldorado National Forest, it does co-occur with its sister taxon, Navarretia prolifera. And the prolifera has more of like a white or pinkish corolla, so they're very easy to tell apart when they're in flower. but absolutely indistinguishable in fruit. So all that meant is that we just had to be really thorough about vetting our populations while they were in flower so that we didn't end up with mixed seed in our seed collections. And I don't know if this is typical for the area where we were collecting seed and the range and the elevation, but certainly in 2024, it felt like winter kind of turned on a dime into summer. And we ended up with a pretty short period of time where things were in bloom and everything was fine. And then very quickly, things were drying out and things were not fine. So we had to pivot. As you guys know, field work happens sometimes, and just kind of rearrange our planning to make sure we were able to get to several populations and confirm that we were going to be able to collect just this subspecies and not mixed seed. Moving on to the Calochortus, the seed collection effort was also quite straightforward. It has these big, beautiful pods that dry out and dehisce naturally. So once the pods were very mature and ready for seed to be collected, we either collected whole pods or we just inverted whole pods directly into envelopes, a very straightforward method. We ended up getting 3 vouchers and about 35,000 seeds from 177 maternal lines. And our only real challenge with this one, which I'm showing in these lower photos here, is for several of the larger populations, which of course were the populations we were targeting for seed collection. When we returned for seed collection, there was quite a bit of herbivory on the mature seed pods. And so that just meant that we had to be really thoughtful and careful about our calculations for not over-collecting. And then we're back to Lewisia. So Lewisia Kelloggii, this seed collection process was very complicated, actually, and there were a lot of unknowns going into this. But that meant that we had a lot to learn, and that's what I'm hoping to share here with you in the last handful of minutes of my presentation. We ended up with 4 vouchers and about 6,500 seeds from 84 maternal lines. And I'll just point out that both of those numbers are an order of magnitude lower than the seed and maternal lines that we were able to collect for the other 2 taxa. But it's frankly a lot more than I expected us to be able to collect, so I still think a huge success. And then with absolutely no drama, I will note that literally everything was challenging about collecting seed for this taxon. So why was it so challenging to collect seed for Lewisia? Well, it disappears after it flowers. It has retractile roots, and what that means is that after anthesis, it does a little jockeying. The big fat fleshy taproot expands and contracts a little and adjusts the position of the whole plant relative to the soil surface. And basically everything that is still remaining above ground, so senescing flower, maturing fruit, some leaf material that's still left over, just sucks right back under the ground. And so what this meant is that we, while we were visiting populations during the flowering period, we had to figure out a way to mark individual plants so that we could relocate them when we came back for seed collection. And so that's what I'm showing here in these photos. These are what I know to be called survey whiskers. They're about 8 inches long, and it's just a little bundle of rigid plastic that's tied together with a metal ring. And then you can use that metal ring to hammer it with a big hefty 6-inch nail into the ground. So that's what we came up with as our method. But we didn't really know how well that was going to work. So here is a photo that I wanted to share with you, or actually a pair of photos, both from Courtney on our team, and I think it was actually a fluke that we managed to get these photos. This photo was taken in late June, and just about a month later, this photo was taken of the exact 2 same plants. And in June, you can see that we have plants there in flower at the base of each of those survey whiskers. And a month later, you couldn't tell that there was ever a plant there. There was not even a divot in the soil. So once we had figured out just that part of how to be able to relocate plants once they had disappeared back under the soil, our next problem was, what are we going to find when we show up? Are the seeds going to be loose in the dirt? Does this plant even have high seed set, right? Or does it abort a lot of its fruits? We just really didn't know what we were going to find when we came back. And so in this photo, I'm showing, this is the tip of a hori-hori. And there's a lot of seed in this picture. Actually, I've only circled a handful of them just to make the point that even though they're fairly large black seeds, in that decomposed granite, they just blend right in. And I have a couple more photos here that show the different conditions in which we found seed when we did come back. So on the far side there, this is a plant that actually still had pretty fleshy sepals. That was very rare. Mostly it was all desiccated and senesced by the time we came back. And then right in the middle, that's a partially desiccated and disintegrated capsule with seed sitting in it. This is another capsule with seed sitting in it, and all of the fleshy parts are gone. In the middle here, this is an entirely intact capsule. So there's just seed in the middle there, and it was actually a totally fluke find. That was really fun to find. And then on the far right here, this is actually probably the most common situation that we encountered, where, I know this is really zoomed in, but this is basically compacted soil that's just stuck to the outside of what's remaining of the plant, kind of around the top of the fleshy taproot, and the seeds are just embedded in it. And so these are all little seeds, and so that's mostly what we found. And then our last big problem was making sure that we could collect this seed and not kill the mother plant, because we're trying to do conservation work. And basically we had a lot of conversations leading up to our effort to talk about tools, methods, process, and how are we gonna accomplish a partial excavation, seed collection, and reburying without killing the mother plant or damaging the tissues. And for fun, as I said, we did voucher plants, so I included this photo just so you can see what an entire plant dug up looks like with that big, beautiful, fleshy taproot. And I have one more slide here. It's actually a video that I'm gonna talk you through that is our seed collection process so that you can see it. And I don't know what making this, great. I will say this is not cinematic greatness. The video is not great, but our process was to return, and we determined that it was actually fairly easy to pull that whisker out, and using a hori-hori to just in shallow lifts, like half-inch lifts, move the soil away across a broad area. And you're probably not gonna be able to see it very well in the video, but right in the middle there, you can see I've honed in on something in the middle, and there's just some desiccated plant material there. That's it. That's mostly what we found. And so then to continue scraping the soil sort of down and away from that, just enough so that we could access the seed. On occasion, when we found a plant that was in a little bit fresher condition, it was possible to actually pluck with your fingers the little capsules right off the top and put it in the envelope, but mostly we used either the pointy tip of a hori-hori or a spoon. And this video is gonna conclude a little short. I cut it short for time, but basically once we get a bunch of the seed collected, not 100%, we pushed that soil back over and matched what the condition was when we found it as best we could. So that was our process. And as I said, this was a huge collaborative effort, and I just wanted to thank everyone that participated. A lot of Forest Service staff helped out. Jordan and Courtney on my team at CNPS were instrumental. A bunch of other CNPS staff also volunteered their time to help with this project. And then we had all of these amazing local people that know the forest way better than I do and contributed their time and driving miles to help make this project a success. And I definitely used at least some of my question time. I don't know if I have time for any. I can take 2. Maybe in the far back there.
Yeah, Kristen, you said that you got the soil back over the plants after done. Of course, we didn't get to see that part of the video, but did you guys try to recreate the texture? It looked like there was a lot of cobble or something there. I was wondering if that actually is important to these plants to have that sort of rough and irregular surface with sort of cobble over it.
Yeah, thanks, Fred. Most of the areas where the Lewissia occurs are these lava caps with really, really compacted soil. It was very hard to pound those survey whiskers into the ground, I will just say that. But yes, we did our best to try and get the fines back and then the cobbles and like actually get it back as best we could find it. And unfortunately, we don't get to go back and track these plants next year. Like individual plants, but all I can say is I feel good about the process, and like we were able to do it without exposing the roots or really damaging any of the root tissue. So just come in delicately, grab the seed, and get out, kind of a thing. And then I think there was a question up front.
That was awesome. Thank you. I was curious if you have any thoughts on like the seed dispersal mechanism or why this plant might be wanting to hold its seed right against the parent.
Yeah, that's a great question that I actually have thought a lot about. And the best that I can come up with is that these are in areas that get snowpack in the winter. So they're mostly around like 8,000 feet elevation. And I think that it must just be that the plants are, the animals that are running around and like digging up that first few inches of soil and run around on the ground, above the ground, but below the snowpack and doing all of that kind of thing are helping with dispersal. And that it must be more important to avoid the desiccation and the heat of late summer. Total hypothesis. I'm open to criticisms on that, but yeah, we talked quite a bit about that, and I'm not really sure what the advantages of like just holding your seed close. Yeah, so I think that's all the time I have. Thank you very much.